Here we see, with scanning electron microscopy as well as transmission electron microscopy, sperm entry into the sea urchin egg. The sperm has come in the vicinity of the egg, and the microvillus of the egg has attracted the attention of the acrosomal process. And in fact, the acrosomal process from the sperm is able to recognize the egg at that point, that the binding particles that are on those microvilli attract the same species of sperm. Here you see the egg is enveloping the anterior part of the sperm head, and it's starting to go into the substance of the egg. And here in panel C, you can see that the sperm has actually begun to penetrate, and the membranes have fused, the sperm membrane and the egg membrane. This is a transmission electron micrograph which shows you the nucleus of the sperm, and it's now beginning to enter the egg cytoplasm, and so the next step will be for this sperm pronucleus to migrate toward the center of the cell, where it will fuse with the egg nucleus to form a zygote and to return the animal to the full diploid condition. Occasionally you get polyspermy, and what happens in this case is that two sperm or two or more sperm may enter the egg. That usually doesn't happen because of the safeguards, but it does occasionally. And certainly things like the salt concentrations of the seawater or other conditions can sometimes cause polyspermy. But basically, when that happens, you end up with an abnormal zygote, because what you end up with are two sets of centrioles, so that you're going to end up with a double set of chromosomal retractors. As soon as the sperm enters the egg, the sperm nucleus then would ordinarily fuse with the egg nucleus, and the sperm microtubules form the mitotic apparatus. Here you see, if you have a double set of centrioles, you will get a double set of apparati that are going to start pulling in a contractile way, pulling the chromosomes to the opposite poles, but you end up now in the first division with 4 cells, and there isn't necessarily an equal number of chromosomes in each of those cells, and so you end up with an embryo maybe dividing for a few cells, but then it's going to disintegrate, because once the embryo reaches a level where it no longer is being controlled by the maternal RNA and proteins and so forth that are put in the egg, but it depends on the chromosomes now for its promotion of proteins and other materials that allow it to continue to divide. Once that happens, then the embryo clearly cannot survive any longer, and it will die. If you look here, what you're seeing here is you've got 2 sets of centrioles with the microtubules pulling chromosomes in opposite directions, but again, this is an actual fluorescent micrograph which shows that the chromosomes are now being pulled in aberrant ways. H is actually a mammalian cell which has polyspermy, and here you get 4 centrioles, 2 sets of centrioles, so the chromosomes, instead of going back to the condition of being diploid, will be randomly numbered as they go to the various blastomeres, and again, they will not be able to continue to develop. This shows one way that the eggs avoid polyspermy. If the sperm is added, and once the sperm fuses with the egg, basically the membrane potential will increase to the point where no more sperm can come into the egg normally. And so when that happens, you get the usual first division with 2 cells that have returned to the diploid condition. If, however, sodium is reduced, then the sodium level is too low. It will not elevate the sodium up to this level to protect the egg from polyspermy, and so you will get multiple sperm that enter the egg, and then when you get the first divisions, you get a random number of cells, usually 4, but you might get even 8 or more than that, and clearly that's incompatible with continuing development, incompatible with life. This also shows the formation of the fertilization envelope, which is to remove excess sperm that have attached initially to that jelly coat and to the vitelline membrane. The formation of the fertilization membrane actually will start on one end where the sperm enters the ovum, and when that happens, it starts to go around fairly rapidly, and it will knock the other sperm off. And so the sperm that initially got into the egg will be the one that will end up fusing with the egg nucleus, and that will be the one that will fertilize the egg. Once the fertilization envelope is complete and the excess sperm have been removed, which is illustrated here, it's really a zygote then. The nuclei fuse and the egg will begin the division process. This shows a diagrammatic as well as an electron microscopic representation of what's happening also in the formation of the fertilization envelope to avoid polyspermy. So there are a lot of things that keep polyspermy from happening. It's a sort of a multi-layered approach to avoiding polyspermy, because clearly if that happens, that's incompatible with life, and that's not going to allow proper development to take place. But here you see when the sperm comes down to join with the microvillus, goes through the egg jelly into the area of the vitelline envelope, and it knows which egg to go to based on the chemical components of that egg that are compatible with that particular species of sperm. And once that touches that point and begins to penetrate into the egg, the cortical granules will start to release their content, and as they release their content, they will pull water into the egg under the fertilization envelope, so it separates then the fertilization envelope from the actual egg cell membrane. The fertilization envelope becomes rigid with the presence of hydrogen peroxide, and that further stops the penetration of the sperm. And so if you look in the electron micrographs, you can see this basically down here. Fertilization has not taken place here, and then once it's fertilized, there is a rupturing of that, the cortical granules, and the fertilization membrane comes into place and stops the entrance of any more sperm. So if you look at the events of fertilization, just very briefly, includes the sperm and egg binding, and that's based on chemical events that allow the sperm to recognize the egg of the proper species. The fertilization potential rises, and there's a real fast block to polyspermy within a second. The first phase, the electrical phase, starts. Then there's a calcium increase. Cortical granules release to elevate that fertilization membrane. And so within a minute or less, it would be impossible for an additional sperm to enter the egg. It just simply couldn't happen. Could happen perhaps if two sperm come at the same time, and that's why you do occasionally get polyspermy. Some of the other responses, of course, is the sperm entry, and that takes 1 to 2 minutes in duration during the 1 to 2 minute period after the sperm has come in contact with the egg. Sperm nucleus then will migrate to the egg center. Egg nucleus will migrate to the sperm nucleus. There's an activation of protein synthesis, amino acid transport, initiation of DNA synthesis, and finally you get mitosis and the first cleavage. If we look at this in a differential interference contrast microscope at various phases of fusion of the sperm and the egg nucleus, you can see that the sperm nucleus is here with the centrioles sending out the microtubular arrays that are going to give rise to the materials that will set up the mitotic spindle. Here's the egg nucleus. They migrate toward each other. Now these are after time lapse, and you can see that eventually they come together. The sperm nucleus, egg nucleus will fuse, and at this point you have a zygote. And here you can see this is with fluorescent microscopy. These are microtubules that have extended out from the area of the centriole that the sperm nucleus brought with it into the egg. Here's the egg nucleus, and they're at about just seconds from now fusing into a single combined male-female pronucleus so that the zygote will be formed. Here's an electron micrograph illustrating as the sperm nucleus and egg nucleus are coming together, about ready again to join. If we look at internal fertilization in mammals, the process is basically the same as the sperm enters the egg, but the process of getting the sperm to the egg is significantly different in the mammalian system. This is a scanning electron micrograph showing a bull sperm as it adheres to some of the membranes in the oviduct of the cow prior to entering the ampulla, where actually the fertilization of the egg takes place by the sperm. As I mentioned, it's really quite difficult, or it has been difficult, to study fertilization in mammals because it's not something that you can set up easily in the laboratory, although now, of course, with some of the studies on in vitro fertilization, a great deal has been learned. You know, of course, the sea urchins, you can set up various experiments, and that's not quite as easy to do in the case of the mammals. It's also difficult in the mammalian system because the sperm in the mammal in a given ejaculate are not all at the same level of differentiation. The spermatozoa are at different levels of differentiation and different stages of maturation. Of the 280 million or so sperm in an average mammalian ejaculate, probably only about 200 of those actually eventually reach the area, even the area of the ovum, because most of them are lost along the way. So that's really probably about 1 in every 10,000 sperm that will be in the vicinity of the ovum, with the opportunity perhaps to fuse with the ovum to end up forming a zygote. And they get there, the sperm actually gets there by multiple mechanisms, as does the egg to the ampulla portion of the fallopian tubes. And this translocation process, the oocyte is moved down the oviduct to the appropriate place, as I mentioned, the ampulla, where fertilization takes place, and much of this movement is probably due to the smooth muscular contraction of the oviduct. The translocation of the sperm, it's often thought that the sperm swims to the point of the ampulla, but that's really not the case. The sperm doesn't have the energy to be able to go that far. It's a very tiny structure, and the translocation of the sperm initially to the ampulla from the vagina may be in part due to the flagellar movements, but mostly by the contraction of the oviduct and the uterine muscles. The uterine muscle contractions are critical in getting the sperm to the oviduct, and continuing onward, the region of the oviduct before the ampulla may actually slow down and sequester sperm so that the sperm is released over a significant period of time. It may be as long as a week that the oviducts will retain the sperm, releasing a certain number at a time to increase the opportunity to get at the egg at the proper time so that fertilization can take place. Of course, once the sperm is in the vicinity of the egg, then flagellar motility becomes very important, because the flagellar motility is what's required to actually penetrate through the jelly coat of the egg, through the vitelline membrane, and through the egg membrane to fuse the egg membrane before releasing the nuclear material into the ovum. The sperm also is influenced by different temperatures in the mammalian oviduct. During the trek from the vagina to the ampulla region, the sperm matures such that it has the capacity to fertilize the egg when they finally meet. And this maturation is called capacitation of the sperm. When the sperm is first ejaculated, most of the sperm, probably none of the sperm, has the ability to actually penetrate an egg, to have an acrosomal reaction, and to actually fertilize the egg. The newly ejaculated sperm has to undergo various kinds of modifications while it's in the oviduct to be able to do that. And that set of physiological changes is called, again, capacitation of the sperm. Capacitation can also be done in vitro, and that's how the in vitro fertilization clinics operate, by treating the sperm prior to the time that it's used for in vitro fertilization, by treating it with various kinds of drugs and so forth to cause that capitation to take place in an artificial situation. Although some sperm may reach the ampulla within 30 minutes coitus, these sperm probably don't have the ability to really fertilize the egg, because they haven't had capacitation The oviduct binds and capacitates the sperm then, and then releases them, as I mentioned before, over a time period in packets of sperm that can prolong the time then that fertilization might have an opportunity to take place. There are various processes that constitute capacitation and enable the sperm to fertilize the egg in mammals. The sperm membrane is altered by removal of cholesterol and albumin proteins by the female. There are changes in the location of lipid rafts, and there are a variety of other changes that allow the sperm to be able to bind to the zona pellucida of the egg and also to be able to participate in the acrosome reaction. Without these changes, like I say, the sperm is pretty much an inert cellular component that really can't participate in this fertilization. The membrane potential of the sperm becomes more negative as potassium ions leave the sperm, and this facilitates fusion. Protein phosphorylation occurs. There are other chaperone proteins that move to the surface of the sperm, and this allows, again, it facilitates the sperm-egg fusion. The outer acrosomal membrane comes in contact with the sperm cell membrane, again, to prepare it for fusion. And getting back to this micrograph again, which obviously is artificially colored, the sperm is pink. The uterine oviduct lining has many ciliary and flagellary kinds of structures. These are extensions of the membranes of the epithelium, and no doubt this is an example of where the sperm is undergoing capacitation in order to be able to fertilize the egg. And I mentioned before there's a thermal gradient that exists between the oviduct and the warmer isthmus where the sperm will swim across that area. So there is an elevated temperature zone as it nears the egg. The other thing that's important is that the cells surrounding the egg secrete progesterone. And so the cells surrounding those oocytes will release progesterone, and that will significantly activate the sperm and make the sperm such that the flagella swims very rapidly and heads toward the ovum, attracted by the progesterone that's in that localized area. And as the sperm comes in the vicinity of the egg, it starts to undergo the acrosome reaction. Initially, in this transmission electron micrograph A, you can see that the acrosome membrane and cell membranes of the sperm are breaking down, and if you look at that diagrammatically, you can see that the sperm cell membrane breaks down into globular units so that it exposes the acrosomal projection, and that acrosomal projection then can penetrate into the membrane of the egg and will allow fusion of the sperm membrane with the egg membrane. Here we see the entry of the sperm into the golden hamster egg, and you can see in figure A that the sperm is going in sideways and it's penetrating and fused with the egg membrane. If you look at the bare area up here, that's where the polar body is forming now that the sperm has initiated the fertilization process. Here you can see it's also entering in through the jelly coat layer, and it's going in sideways. It goes in sideways. This is the egg membrane, and it is in the process of fusing. Here is the acrosomal projection right here that is entered in, and here you can see this diagrammatically. And again, the sperm approaches the ovum from a side viewpoint. It will be engulfed, in a sense, by the egg, and it will fuse this way. The sperm nucleus will then migrate toward the egg nucleus, and there will be a fertilization to form the zygote taking place. If we look at human fertilization, again, the pronuclear movements during human fertilization, you can see that the microtubules are stained green, DNA is blue. A is an unfertilized ovum, so it's mature but it's unfertilized, so you see there the first meiotic division. It's an oocyte up in this area here. The sperm has entered the egg right here now, and you can see that there are a number of microtubules extending out from where the sperm entered the egg, and here the polar body has divided now to complete meiosis, And the sperm nucleus then will continue to migrate toward the egg nucleus. Here's the tail of the sperm still evident. Here's the egg nucleus, sperm nucleus, and they will fuse together. And here you get the first mitotic division. Still seeing the sperm tail present. Here are the centrioles that were brought in by the sperm to form the mitotic spindle. And here again are the blue stained DNA. And you can see that the chromosomes then are about ready to be pulled apart to form the first two-cell stage and to really begin the formation of the human embryo. So this pretty much completes what I wanted to talk about regarding fertilization. It's a complex process. It's much more involved as you get into it than one might originally think. There are differences among the invertebrates and vertebrates, mammals, and even among the different mammals. However, there are some things that remain in common that make it possible then to return from a haploid state to a diploid state and complete the genome so that development can continue and so that a new organism can form.